So in our last video, we talked about polymerization, and in the video before that, we talked about the chemistry of carbon. And today, we're going to delve into the specifics of those four biological macromolecules, biopolymers, that basically make up your body. These are the things that your body is made out of, this and water, basically. There's a few other things in there. There's some ions, there's, some mineral elements, there's some other organic molecules. But for the most part, your body, the dry weight of your body, the non-water weight of your body, is almost entirely made out of these biological macromolecules. Remember, there's four of them. There is carbohydrates, proteins, lipids, and nucleic acids. I'm going to go through each of the four categories and tell you about the properties of each. So the first one to talk about is carbohydrates. So the general properties of carbohydrates: it's a polymer, right? That polymer is made out of monomers, and those monomers are called sugars. A sugar is a monomer of a carbohydrate. Monosaccharides is another term that you will hear for it. And there are basically three monosaccharides that your body uses on a regular basis: glucose, fructose, and galactose. Also ribose, which I'll get into in a second. I also call it four of them. The most common one to be used in your body to make polysaccharides, carbohydrates out of, is glucose. Glucose is the main carbohydrate monomer used by your cells. Polymers of glucose come in several varieties. Starch, which is used by plants, is a polymer of glucose. Glycogen, which is used mostly by animals, is also a polymer of glucose. Cellulose, this is the material that makes up the cell walls of plants. So that means that it's basically what wood is made out of. That is also a polymer of glucose. Now, both starch, which you can think of as being basically like French fries, and cellulose, which are wood, is made of the same thing. They're both made of glucose, polymerized. But wood and starch, very, very different things. Starch is yummy fried. Wood isn't going to taste very good no matter how you prepare it. So how you put them together, how you arrange the molecules, how you connect them to each other, can have a huge difference on the structure of the carbohydrate that you make. In terms of the structure of carbohydrates, the monomers, your basic sugar monitor, are made of carbon, hydrogen, and oxygen. And that's in the name. Carbohydrate, right? Hydrogen for the hydra. Anything with an 8 in it, anytime you see 8, that means it has phosphate, or that means it has oxygen. So for instance, phosphate is phosphorus and oxygen. Carbonate is carbon and oxygen. Carbohydrate is carbon, hydrogen, and oxygen. True carbohydrates have equal amounts of carbon and oxygen and twice as much hydrogen. So their general formula is some multiple of CH2O, carbon plus water. Glucose, for instance, as I said, the the carbohydrate that is used most commonly, the monomer that is used most commonly by your body, its chemical formula is actually C6H12O6. Right? So the same amount of carbon and oxygen and twice as much hydrogen. The chemical properties of carbohydrates, they're hydrophilic, they combine well with water, and they are energy rich, which is a lot of what your body uses them for. In terms of naming conventions, carbohydrate names are often built from saccharide or have the suffix "-ose". Both of them mean sweet or sugar. So for instance, polysaccharide means many sugars. Glucose, fructose, maltose, dextrose, those are all different types of sugars. And so they have the suffix "-ose" on them. You will also often see gly or glue in the names of things that are carbohydrates or that have carbohydrates in them. For instance, a peptidoglycan is a protein with sugar groups on it. And peptid means sugar. We'll get to that in a second. That glycan means gly, sugar. Anytime you see gly, glue, saccharide, or ose, you're probably talking about some sort of carbohydrate. How are carbohydrates used in the body? Well, the main way that they're used are energy. All right. So sugar is the body's main source of energy. They're used for storage, typically storage of energy. So your body can burn sugar to make energy that it will use, or it can store that sugar in the form of starch if you're a plant, or glycogen if you're an animal. So you store energy in the form of carbohydrates as well. Carbohydrates are also used for structure. You can build things in the body that are going to give your body shape. This is particularly true if you're a plant. Cellulose, the carbohydrate, cellulose, which is a polymer of glucose, is used by plants for shape and structure. Animals do not typically use a lot of carbohydrates for their structure, but there are carbohydrates that form key components of what's called the extracellular matrix, which will be a topic of our discussion in a few weeks. But to give you the super basic version, the extracellular matrix is what holds your cells together and gives shape to your cells so that they're not just a mound of tissue, right? If it wasn't for this extracellular matrix, you would just be a blob of tissue with no organization. Last, carbohydrates are used for recognition. You've probably heard of different blood types. We're going to talk about those more extensively when we talk about blood in the next semester. But the way your body recognizes, oh, this is my blood versus that is somebody else's blood that I need to destroy, that, you know, this is type A blood or type B blood or whatever, is because of recognition elements on the surface of the cell. And those recognition elements are often carbohydrates. It's how your body tags cells to label them as being things that belong and to communicate where they belong so that your body can tell, oh, this is a blood cell or this is a liver cell. And if it gets in the wrong place, it might destroy that thing because that means that it's, you know, potentially becoming cancerous. So your body labels cells with carbohydrates so that it can recognize its own cells. Disaccharides, remember, these are polymers. They're made from monosaccharides. So glucose is a common monosaccharide. You put two glucoses together, you get maltose. You put glucose and fructose together, and you get sucrose, which is a common table sugar. Both of those are put together using dehydration synthesis reactions. And the linkage, the bond that links the two sugars together, is called a glycosidic linkage. Remember, glyco means sugar, so basically that just means a sugar linkage. And we can label different types of sugar linkages, and some of them are easy to digest and some of them are less easy to digest. Longer polymers, polymers with many hundreds or thousands of glucose in them, which is how you store glucose, are usually going to be either starches or glycogens, or sometimes celluloses. I'm not going to get into the structure of cellulose here, but starches are an amylose structure, and they basically are a linear chain of glucose in a spiral. So you've got one long spiral of glucose. Some plants will have a few branch points. A branch point in a starch is called an amylopectin. You may have heard of pectins as a thing that you find particularly in fruits. But these branch points are fairly rare. So for every molecule of starch, usually you're going to have one starting end and one or a few ending ends. Glycogen, on the other hand, has many branch points. So you can see this glycogen has a branch point here, and then there's another branch point here, and another branch point here, and another branch point here. So you might start a glycogen with just one chain, but by the time it branches off and branches off and branches off, you might have many, many, many ending points. This is actually something that's really important to how plants and animals are different. Right? Plants store their glucose in big long chains because long chains pack well. You can store them very, very efficiently. It's like a bunch of equal-sized boxes. You can pack a whole bunch of them into a small storage area. But just like if you have a bunch of boxes in a storage area, you can only get at the ones at the front. When you're freeing up energy, you can only free up energy from the ends, one at a time. So this plant, how much energy can it get at? Well, it can only get at one glucose at a time from the polymer that it makes. If it's got some amylopectins, then it might be able to get at 2 or 3 glucoses, but it's not going to be very many. For plants, that's fine. Plants have very predictable energy needs, right? During the daytime, they're using pretty much the same amount of energy all the time. During the nighttime, they're using pretty much the same amount of energy. There's a bit of a difference between day and night, but plants don't have varying energy needs, right? If a vicious rabbit comes by and decides that it wants to eat the carrot doesn't have a whole lot that it can do about that. It can't get up and run away. It doesn't need to hunt anything down. Pretty much it just bees a carrot. So if it gets caught, it gets caught. Animals have very different energy needs, right? When you're sitting there in front of the couch, you don't need a whole lot of energy. On the other hand, a bear, crashes through your window, goes "Rawr!" in front of the TV. You need to get up and run away. Now you need a lot of energy really, really quickly. Because every molecule of glycogen has many ends on it, you can get at a lot of that sugar much more quickly, right? You can be releasing, 5 or 6 or 7 sugars at a time instead of just one. And actually, real glycogen would have many more branch points than this because it would be much longer. Now, that has some problems. That means it doesn't pack as well. So you can't put as much in a small space, but you can get at it much easier, and you can stash it away much easier as well. So animals usually get their energy in a big meal, right? Maybe 2 or 3 times a day. And you've got to be able to grab the sugar out of your bloodstream and stuff it onto the glycogen really, really quickly and get it out of the blood. Plants constantly make sugar throughout the entire day while the sun is shining. So they have a very steady drip, drip, drip supply of sugar that they need to pack away for storage. Animals are likely to get their glucose in a big lump sum. They have to pack it away really quickly, and then they might not need it for a while, and then they have to get at it really quickly. So for that reason, glycogen has these many, many ends and this branched structure. This is actually a great example of structure-function relationships. And this is something that you're going to have to pay attention to as we go through this course. I'm going to even write it out here. Structure, function. Because animals and plants have very different functions that they need to perform, they store their glucose in different structures. You see how the structure of starch or amylose is going to provide advantages for the plant that suits its lifestyle, whereas the very different structure of glycogen is going to provide advantages to the animal in terms of ease of access, rapidity of storage, things like that, that suit its lifestyle. So the structure of the molecule determines what function it is used for. Our second molecule that we're going to talk about is lipids. All right? Lipids are either polymers or monomers, depending upon the type of lipid that we're talking about, but they're not macropolymers, right? So a molecule of starch might have thousands and thousands of monomers in it. A protein might have hundreds or thousands of amino acids in it. A DNA strand could have hundreds of thousands of nucleotides in it. For lipids, it's usually going to be like 1, 2, or 3, and it doesn't really get more than that. There are 2 types of monomers that you might see in lipids, and those are fatty acids and steroids. This is a fatty acid. A fatty acid is a long chain of carbons with a hydrogen on each of the carbons. And then at the end, you can see here, we have a carboxylic acid group, and this right here is the acidic hydrogen. And so we have a long chain of hydrophobic, what are called hydrocarbons, just carbon and hydrogen, and then a little bit of an acidic end. That's why it's called a fatty acid. This is a steroid molecule. You can see that it has this structure which is based around a set of 4 rings, 3 hexamer rings and 1 pentamer ring. Hexamer means 6-sided, pentamer means 5-sided, and then you will often have this little tail kind of curling away. This is cholesterol, which is the thing that all steroids are modeled after. And you could have different functional groups hanging off of different areas here that make it into different types of steroids. And similarly, your fatty acids might have different numbers of carbons in them. So this is palmitic acid, but you could have shorter or longer fatty acids. This is what's called a saturated fatty acid. That means it contains all single bonds. You could have an unsaturated fatty acid, which means that it contains at least 1 carbon-carbon double bond. Monounsaturated fatty acids, you probably have heard of these, monounsaturated fat, polyunsaturated fat, saturated fat. Saturated means it's got all single bonds. They're the most energy rich. Monounsaturated have one double bond. Polyunsaturated have more than one double bond. So those are the two structures, and they're usually pretty simple. The structure, as I just said, fats are made of these long hydrocarbon chains. It's important to point out that these are just single chains. They're never branched, right? Just linear chains. That's it. Steroids have this quadruple ring structure. In terms of the chemical properties, lipids are defined as the hydrophobic molecules. Of the four hydrophobic, of the four biological macromolecules, lipids are the only ones that are not water soluble. They do not dissolve readily in water. They are very hydrophobic. So they are said to be soluble in organic solvents, which is going to be things that are hydrophobic. How are lipids used in the body? Well, the main one that everyone knows is energy storage. Didn't we just talk about how your body stores sugar in the form of glycogen for energy storage? Well, we also talked about how glycogen is specifically designed, specifically made so that it has all of these ends where you can release sugar really, really quickly. Glycogen is how your body stores energy short-term, energy that it might need to get at really quickly. That's the first source of energy that it uses. That's like energy that is stored, in your garage or your basement. It's really easy to go get that. Lipids are long-term storage. If you want to use your lipids for energy, you have to go get them from your fat cells, use a special process to break them down, and then usually convert them into glucose before you're going to use them. So, but they're really, really energy dense. So you can store a lot of energy in not very much space. So this is like your storage warehouse, all right? Takes a little bit longer to get at it, but it's much more efficient for storing energy. Protection. Fat is soft and squishy. Your body uses fat to surround many of its organs, and it's basically like bubble wrap for your body's organs. It protects them, keeps them from getting damaged, and it also protects them from temperature shock. Fat is insulating, right? It holds in heat, and it also holds out heat. But for your body, mostly it's going to hold in heat so that when you step out of doors in, January, you don't just freeze to death, right? That fat layer around your organs and underneath your skin is going to prevent heat from leaving your body too quickly. There are some lipids, special lipids called phospholipids that we're going to talk about pretty soon when we talk about the cell. These phospholipids are what makes up the membrane, which is like the skin of cells. They make up the thing that separates the inside of the cell from the outside of the cell. And there are some special things about the structure of phospholipids that we'll talk about in a bit that allow this to happen. So you use lipids, these special phospholipids, to build cell membranes. Last, but by no means least, is control. Right? This is where steroids come in. Steroids are used for both structure and control. Fatty acids are used for energy storage, protection, and structure. So you'll find cholesterol in the membranes of your cells as well. They play a role there. They're essential to have. You die without enough cholesterol. But you also take cholesterol and you make hormones out of it. So steroids are one class of common hormones. And hormones are things that your body uses to control and coordinate its actions. Some other just examples of places where you're going to see lipids used in the body. Bile acids, these are things that help your body to digest fats. Cholesterol, which I just said has a role in membrane structure. Conosoids, which are hormone-like things. They're chemical messengers that send information between cells. There are some vitamins that are basically lipids or are made from lipids, specifically vitamins A, D, E, and K. These are essential for your body to do its functions. They are very important, and they are fat soluble, and they're made from lipids. Fatty acids, we just talked about them. Phospholipids, those very important lipids that make up the membrane of cells. Steroid hormones, chemical messengers based off of cholesterol, and triglycerides. Triglycerides are 3 fatty acids. So a triglyceride is basically a carbon, 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 and then it's got a fatty acid hanging from each of those carbons. So it's 3 fatty acids linked together with these carbons, hence triglyceride. This is the form that your body stores fat in for energy storage and for like thermal insulation and protection. When you think about fat, right? When you poke your body and you go, oh, there's some fat there. That's triglycerides. I do want to make a note. You've probably heard of like good cholesterol versus bad cholesterol. Some of you may think cholesterol's like, really bad. You don't want to have it. Some of you might think that, oh, there's this difference between good cholesterol and bad cholesterol. There's actually only one type of cholesterol. It's cholesterol. There's only one. There's no good. There's no bad. And in general, in your body, it does more good than it does harm. You have to have it. If you don't have it, you die. What this good and bad cholesterol refers to is actually because cholesterol's a lipid, it's not water soluble. Well, your blood is mostly water. So how do you move cholesterol around? If the cholesterol doesn't want to get into your blood, how do you move it around your body? Because the blood is what moves things around your body. And the answer is that you have these special proteins, and those proteins are called lipoproteins. So lipoproteins, all right? These are proteins that basically what they do is they bind to the cholesterol and they protect it from the water so that the cholesterol can move around your body, and it's like shielded by the protein, and the protein is shepherds it around the body. You can think of it as being like a bus that's going to protect the cholesterol from the water as it moves around the body. These lipoproteins, they bind cholesterol as well as fats and phospholipids to move them around the body, because again, fats and phospholipids can't move on their own. There are multiple types of these lipoproteins. Actually, there's only a few types, but HDL, high density lipoproteins, are what are thought of as the good cholesterol. They're not actually different proteins, but think of this as like, this is a bus that has a lot of empty seats on it. Okay? It's got some cholesterol in there sitting in the seats, but it's got a lot of empty seats. And so that means that more people can get on the bus and people can get off the bus, right? So they have a lower ratio of lipid to protein. They've got a bunch of empty seats, not too much lipid. These can actually pick up random cholesterol that's floating around. So if you have cholesterol that's beginning to build up in a place and starting to turn pathogenic, to turn to turn atherosclerotic or start to do damage, the HDL can actually pick them up and give them seats on the bus. LDL is a bus that's got most of its seats full. It's already got cholesterol sitting in all of its seats, and more of that cholesterol is leaving the bus. So they have a high ratio of lipid to protein. They've got lots of lipids, not a whole lot of protein, and that means that they aren't going to suck up any free cholesterol that might be conglomerating in some place, and in fact, they're often going to dump off a bunch of cholesterol. And that cholesterol, since it doesn't want to be in the water, will start to conglomerate together into these large plaques, and those plaques can do damage to arteries and the heart. So your HDL versus LDL isn't necessarily there's good cholesterol or bad cholesterol. It's a measure of how good your body is doing at moving the cholesterol and managing the cholesterol that it has. But when we measure cholesterol, these are what we measure. Okay, so our third polymer, biopolymer, is proteins. Proteins are probably, I mean, it's difficult to say what the most important is. They're all absolutely essential for life, but proteins are probably the most versatile, the most used, and to a certain extent, the most complicated. We're going to talk about proteins very extensively later on in this course. To give you some general ideas of the properties of proteins, the monomer of proteins is an amino acid. And down here you can see several different amino acids. And I'll talk about their structure in just a second. The polymer is called a polypeptide or a protein. Generally speaking, a polypeptide is shorter, smaller. If you have like less than 80 or so amino acids together, we usually call that a polypeptide. If it's more than 80, we usually call that a protein. The technical definition is a little bit more complicated than that, but that'll get you by right here. As far as the structure of the chemical, all amino acids, almost all amino acids, are based around a similar structure, and it's very easy. On one side, so like right here, see that NH2 group? That is called an amino. Group, or an amine group. This over here on this side, that is a carboxylic BOXYLIC acid. And this carboxylic acid we've talked about before. So you know that you've got your amino and your acid. So hence it's an amino acid. In the middle is a linker carbon. All right. And then off of that linker carbon on one side is a hydrogen. And on the other side is what's called an R group. So this right here is the R group. And note that all of these amino acids that you see here, everything is the same except the R group. The R group is a variable. There are 20 different amino acids that your body uses, and each one has a different R group. So all amino acids have the same amino and acid part, and that's how they polymerize, that's how they hook together. But each R group is different. And so some R groups are nonpolar. So this R group here, this is methionine. It has a fairly nonpolar R group. This is mostly nonpolar bonds. This is a polar R group right here. This is a cysteine, and this SH here is somewhat polar. Whereas down here, this tyrosine, right? So this is an interesting one because this group here in the middle, this region here, is very, very nonpolar. But it's got this OH here at the end that can form hydrogen bonds. So you can have hydrogen bond forming portions in the R group. And this over here, this is arginine. Arginine is also polar, and not just polar, it's also charged. This is actually a basic amino acid, and so it can act as a base. So the amino acids can be acidic, basic, charged, polar, non-polar, hydrogen bond forming, non-hydrogen bond forming. They can be big, they can be small. This is what allows proteins to have so much usefulness, so much variety inside of the body, is that there's 20 different chemical properties that can be attached to these different R groups, and that's what gives proteins all of their power. In terms of how proteins are used in the body, the primary way that we're going to talk about is chemistry. So about half the proteins-ish used in your body are used to make enzymes. And an enzyme is a biological macromolecule, typically a protein, but not always, that can perform a chemical reaction. These chemical reactions are what allow you to be alive. They're what allow your body and your cells to do stuff. Everything that happens in your body happens because enzymes make them happen. But that isn't the only way that proteins are used. About half the proteins in the body are also used for structure. So many proteins, some of which you may have talked about already, very briefly, go into making up the structure of the body. So particularly collagen. Collagen is a tough rope-like protein that actually has a tensile strength stronger than steel. And it is what makes up most of your body that isn't made out of cells. So a lot of your tissues contain these collagen ropes that are going to bind things together. They're what makes up your cartilages. They make up a large portion of your bones. They make up your tendons and your ligaments. And not only that, but they're what gives your skin its ability to resist damage, and they're what organizes your organs. They're what gives it the ability to have complex layers of cells. Movement, all right? So when you move your body around, it's your muscles that move your body around, and you've probably heard that muscles are very protein-rich structures. Specifically, they contain enzyme and structural proteins called actin and myosin, which we will talk about a lot when we talk about muscles, but to give you the brief version, these are what are going to move and cause your muscles to contract. So these create contractions. They allow your muscles to contract. Your muscles contracting is what allows you to move your body around. In addition, Actin and myosin in non-muscle cells are what gives the cell its shape and structure and allows components of the cells to move around inside. They are part of what is called the cytoskeleton, a topic that we'll be talking about soon. Control. So, many hormones in the body, these are chemical signals that allow your body to control and coordinate actions. Many of these hormones are either made of proteins, amino acid polymers, either proteins or polypeptides, or they are derivative of amino acids. You take an amino acid and you tweak it a bit, and you can use that as a signaling agent. Your body does that because it's got a lot of amino acids around already, and if you're going to be making a lot of signaling molecules, you want to make them from something that you already have a lot of. If you have to go out and get a bunch of special components, it's going to be hard to make them quickly. Last, energy. Proteins are not the preferred energy source of your body for the most part, but your body can break proteins down and extract energy from them. And it will do so if its preferred energy source, glucose, is unavailable. Protein polymers are hooked together with what's called a peptide bond. And peptide bonds are formed through dehydration synthesis. So this here, that OH there, and this H here are what are going to leave through dehydration and become H2O. So they leave together, become H2O. And the bond that is formed between them is right here, right? That's what you put together. This carbonyl group and this nitrogen NH group, and that creates a peptide bond, and this is the peptide bond. It's this carbonyl bonded to a nitrogen. And the peptide bond is, relatively speaking at least, a very strong and stable bond. It's a tougher bond to break, so proteins tend to be pretty hardy within the body. Now, I said that one of the reasons why proteins are so diverse, so capable of doing a lot of things, is because of the variety of different R groups, 20 different R groups, but I didn't quite explain that in detail, and I want to get a little bit more detailed in this. The other reason, and it's related, but the other reason why proteins are so versatile is because they can assume a whole bunch of different shapes. Most of the other macromolecules have like just one shape. Glycogen looks like glycogen. All glycogen looks pretty much the same. Lipids look like lipids. They all pretty much look the same. DNA looks like a double helix. We'll get to that in a second, but it always looks like a double helix. Proteins can look like a whole bunch of different things. I want you to think for a second about your clothes, all right? Actually, just take a look at what you happen to be wearing. It's probably made of thread, unless you're wearing leather or some sort of, clear plastic polymer or something like that. It's probably made of some sort of thread. And think about the complex shape, like the clothes that you're wearing fit your body. And there are a whole bunch of different clothes that you can go out and get. There's many, many different styles. They fit a whole bunch of different body shapes. They're all made out of the same thread. But that thread has been woven together to form different fabrics, and then those fabrics have been cut and sewn into different shapes, and then you have different items of clothes that you can mix and match together to create different outfits. Proteins are similar to that. They're organized on multiple levels. And I'm going to talk about 4 different types of structure here that you should need to know at least a little bit about. The first is primary structure. The primary structure is like the thread. This is the linear structure of the protein. So like everything that I've told you about thus far, if you were to just say you've got 20 different amino acids, this is amino acid A, B, C, D, and what order are they in? It's in like A, C, D, A, F, G, A, G, G, S, T, P, that sort of thing. The linear arrangement of the amino acids. All right? This linear arrangement of the amino acids is what is eventually going to determine all of the other levels of structure. But by itself, this primary structure is determined by covalent bonds. What amino acid is bonded to what amino acid with covalent bonds? The secondary structure is where you have your string is then used to form a fabric, right? So think of like maybe you're wearing a sweater. Like if you've ever unraveled a sweater or had a sweater unravel on you, you know that the whole thing is made from just one long strand of yarn. Maybe some of you do some knitting or crochet. You might know this. That one strand of yarn, however, is made to assume the shape of a fabric by having it basically, pulled next to and woven in and through other portions of the yarn. And so this one long strand of protein can form 2 types of what are called secondary structures. The first type of secondary structure is called an alpha helix, and you can see it here. This is where one strand is going to wrap up in a spiral. And that spiral shape is formed by hydrogen bonds between the backbone carbons, the backbone materials. So remember, we said that all amino acids have that amino group, the acid group, and the linker carbon, right? And the R groups can be one of 20 different things. These are that backbone is the amino, the acid, and the linker carbon, right? And what you get is hydrogen bonds between the carbonyl group here and the nitrogen group here. And those hydrogen bonds are what are going to form, cause it to form into this spiral here, which you can think of as being like a bar, a rod. It's a strong structure. The other thing that can happen is you can have 2 separate strands that lie next to each other and that form what is called a sheet or a beta sheet, where you have one strand that's going this way and then another strand that's going this way. And then you could actually have a third and a fourth strand. You could have as many strands as you want. They're all going opposite directions, and they're all making hydrogen bonds with each other, forming this broad, flat sheet. And these are all the same strand, wrapping back on each other. Maybe it turns here. Maybe it turns here. Maybe here it actually goes off and does something else for a while before wrapping back around and forming the next strand of the sheet. But this is like the fabric layer. It's where you take your string and you use it to make some fabric, right? You take the string and you fold it back on itself to make structures. But you don't just wear fabric unless you're, Greek and wearing a toga or something like that, right? We're going to take that fabric and we're going to make a 3-dimensional piece of clothing out of it. that is what is called the tertiary structure. The tertiary structure is where you take all of these secondary structure elements, helices, sheets, other helices, other sheets, and then you start folding them. Like if you were to take a piece of paper and fold it into origami, or if you were to take a piece of fabric and start folding it into clothes, and then you staple it together or sew it together along certain seams, and you end up with this very complicated three-dimensional shape that is unique. And every protein, every different type of protein has a different unique tertiary structure. Now, so far we've had primary structure, and we said this was determined by covalent bonds between the amino acids, those peptide bonds. Secondary structure, this is determined by hydrogen bonds between the backbones, not the R groups. This tertiary structure, it is determined by interactions between the R groups, right? And there's a bunch of different types of interactions. Like you might have part of it, maybe this R group over here is positively charged, and this R group over here is negatively charged. And so they have a charge-charge interaction. And so maybe that's holding them together. Maybe like right here we have an R group that is like a hydrogen bond former, and maybe it's forming a hydrogen bond with that R group there. So you have a hydrogen bond holding them together. You can have salt bridges, which are charge interactions, hydrogen bonds, but the major thing that's actually holding this tertiary structure together is something called hydrophobic collapse. What hydrophobic collapse means is that all of the inside portions, all of the hydrophobic amino acids will want to get away from the water. And so when the protein folds, it stuffs all of the hydrophobic amino acids in the middle where the water can't see them. And all of the hydrophilic amino acids are around the outside where they can form interactions with water. And that's the main force that drives this tertiary structure. This is like a garment of clothing. You've taken your fabric and you have you've sometimes cut it, sewn it, formed it into a unique three-dimensional garment that is not necessarily connected to any other garment. Our last story, and I should emphasize, pretty much all proteins have primary structure, they all have secondary structure, they all have tertiary structure. Quaternary structure is optional. Some proteins have it, some proteins don't. Quaternary structure is like constructing an outfit. Some outfits might be constructed of a single garment. You could have like a dress is one garment, you put it on and it's an outfit. But most outfits, you're going to have more than one garment. You know, you're at least probably going to be wearing some underwear, probably some socks. You might have a shirt, and you might have a jacket on over the shirt. You might have some pants on. Maybe you're really fancy snazzy and you throw a hat and some gloves or something like that in there. But this is where you have multiple folded tertiary proteins, and they come together to form a unit. So just like you would put multiple garments together to form an outfit, here we have this that you see here in the gold. That's one protein, one polypeptide chain. This that you see here in the purple, that is a different polypeptide chain. They're not connected. There's no covalent bond between them, but they are bound to each other. They're stuck to each other through the same type of forces that we talked about with tertiary structures. Salt bridges, hydrogen bonds, and hydrophobic interactions. And so what you see here, this is hemoglobin that we have here as the example, and it has 2 beta chains, which are these gold things, and 2 alpha chains, which are the purple things, and that each of those is going to bind to the other 3, comes together to make what's called a tetramer, meaning it's made out of 4 things, 2 betas, 2 alphas, and those all come together to form one hemoglobin molecule. It's 4 different polypeptide chains, but when they are associated together, it all acts as a single unit, as one coordinated outfit. The unique 3-dimensional shape of a protein, that tertiary structure, is what is referred to as a protein's conformation. Its special 3-dimensional shape, its finished form. And we know that form and function, particularly with proteins, are intimately related. The reason why a protein can do the thing that it does is because of its 3-dimensional shape. Some proteins can actually change their 3-dimensional shape, and this is called a conformational change. Often they will have perhaps a functional form and a non-functional form. And you can force a conformational change through binding them to something or through attaching something to them. And they might change from an active form that actually does something to an inactive form. So you can turn the protein on and off through this conformational change, this shape change. A good example of this is in muscle contraction. So the proteins involved in muscle contraction go through a shape change when they bind calcium. We'll talk about this more when we talk about muscles. When they bind calcium, they go through this shape change, and that shape change causes this little, you've basically got this little, all of these little doodads that are bound to this big long string. And when they bind calcium, they all do this, and they move like that. And that forces movement in this direction. That's an oversimplification, as we'll see, but that's basically what happens, and that conformational change on calcium binding is what's going to allow your muscles to move. Since proteins' 3-dimensional structure is absolutely essential for their function, that means that if you unfold a protein in a way that it's not meant to unfold, you've destroyed that protein's function, and that is called denaturation, right? So the natural form of the protein is the form that it functions in, its normal form. Denaturation means that you are taking away its natural form. You're changing it to an unnatural form. Most proteins, once they have been denatured past a certain point, cannot refold themselves back to a functional state. Some of them can be fixed under some circumstances by other proteins called chaperones that will come by and fix them. But usually what happens is you just destroy that protein and then you make a new protein, right? What sorts of things are going to cause denaturation? Temperature, pH, chemical modification, salt, drying, and hydrophobic solvents are the things that are likely to denature a protein, right? With temperature, if you heat up protein too much, heat is motion on a molecular level. So the more you heat it, the more the protein moves. If it moves too much, it moves out of its normal shape, and then it can't get back into its normal shape. pH. Now, remember with pH, what you're doing is you're changing the protonation state. If you lower pH, you're going to be adding protons to it. That's adding positive charge. If you raise the pH, make it more basic, then you're going to be removing protons from it. That is basically making it less positive and therefore more negative. Remember, we said that this three-dimensional structure is partially held together by these charge-charge interactions. Well, if something goes from having a, positive, negative charge interaction that's like holding this thing together to having a positive interaction, now it's forcing them apart. And that's going to change the shape. Chemical modification, that would be like cutting a protein bond, a peptide bond, then you're changing the primary structure, and that, of course, is going to change everything downstream. High salt. So with high salt, it would be like you've got this positive-negative interaction, but if you take this positive charge and you surround it with a whole bunch of salt, negative charges, now it doesn't have any incentive to associate with the negative charge it used to be around. You can maybe put a bunch of positive charges around that. Now they don't need to stick together so much. And that staple, that seam that was holding the protein together, now comes apart. And when you start having seams that come apart on your clothes start to unravel. Drying. So remember I said that hydrophobic forces are part of what holds tertiary structure together. Well, if you don't have water, then there's nothing for those hydrophobic portions to fear, and there's no force that's forcing them to the middle on the inside. So if you dry a protein, often you will destroy its structure, because if it's primarily hydrophobic forces holding it together, you've removed those forces. And hydrophobic solvents, these would be like most proteins are made to work in water, because that's what your body is mostly made out of, is water. All right? Water exerts a hydrophobic force on hydrophobic portions of the protein. But if you put that protein in something that is more hydrophobic than water, like, say, alcohol or acetone or something like that, then the protein will now, its hydrophobic portions will not need to be on the inside. And perhaps you've even made it so that they prefer to be on the outside. And that's going to change the protein. It's going to flip it inside out. This is how, or part of how alcohol is a disinfecting agent. Right? You put alcohol in a skin before you inject a needle, and that's because it tends to kill bacteria living on the skin. How does it kill those bacteria? It denatures their proteins. And that's part of how it works, is that by denaturing their proteins. Once your proteins are denatured, you're dead. As I noted, denaturation is usually permanent. There are some special chaperone proteins that can sometimes help to refold proteins in emergency situations, but usually a denatured protein is just dead. Not only dead, it's actually actively gumming up the works. It's like a broken down car on the highway, right? It's not going to start working again, and it's impeding traffic. The only thing you can do is haul it off the side of the road, get rid of it, take it apart. An example of denaturation is cooking an egg. Everyone here has probably seen eggs cooked. Here's something that sort of troubled me a lot when I was a kid, and it says a lot more about me than anything else, but I'd always been told, well, when you heat things up, they melt, and when you cool them down, they freeze. When things melt, they go from being a solid to being a liquid. But you take eggs are a liquid, you put them in a pan and you heat them up, and what do they become? They become solid. How crazy is that? What's happening? Well, it turns out eggs are very rich in protein. And normally, these proteins that your eggs are made out of, they have all of their hydrophobic bits on the inside, and all of their hydrophilic bits on the outside. Right? And there's a whole bunch of them, all with their hydrophobic bits on the inside, and their hydrophilic bits on the outside. And that makes sense because they're all floating around in water that wants to associate with the hydrophilic bits. And you have all of these proteins, basically a colloid in solution. If you then heat this up, which is what happens when you make an egg, when you cook an egg, you denature the proteins. And when you denature them, now the hydrophobic bits are just randomly scattered around. And so they'll have hydrophobic bits, some hydrophilic bits, and some hydrophobic bits. And it's just sort of random. All right? So the hydrophobic bits are now no longer the parts inside. And what's going to happen to them? Well, these hydrophobic bits right here, do they want to be in contact with the water? Well, there's water all around them, right? And they don't want to be in contact with that water, so what are they going to do? Well, they can't refold properly, but what they can do is stick to each other. And so you get a conglomeration of proteins where their hydrophobic bits will stick to each other. So here we have a protein here with its hydrophobic bit sticking to this protein's hydrophobic bit. And then the hydrophobic bit up here stuck to another hydrophobic bit of a protein. And you get these big conglomerations of thousands and thousands and thousands, millions of proteins all hooked together because their hydrophobic bits are on the outside and they now want to stick to each other. They become sticky. They form these huge agglutinations that then fall out of solution and become solid. And that's what an egg is. That's how an egg becomes solid. When you cook it, you're denaturing the proteins, and that is causing the proteins to all stick together. By the way, this is also how cheese is made. When you take milk, cheese is made from milk, and the way it's made is that you allow bacteria to ferment the lactose into lactic acid. What have you done? You've changed the pH of the solution. You've made it more acidic. That acid causes the proteins to denature, and then once those proteins denature, they stick together, and that's the cheese curd falling out of solution. What you left behind with is the whey, which is a different protein that is more pH tolerant. All right, so when you make cheese, you change the pH. You also usually add a few other things like heat. Heat helps it denature as well, and sometimes you add an enzyme called rennet that actually chemically modifies the protein. It actually cuts it a bit, causing it to fall apart even faster. And so those are examples of protein denaturation that can happen anytime. You can go to the store and you can watch happen in your fridge. Enzymes are going to be really important for one of our future topics, that's metabolism. And I wanted to talk just a little bit about what enzymes are and how they're named and how we talk about them. Remember, enzymes are proteins, usually proteins, that function as catalysts, meaning that they perform a chemical reaction in the body. They have a natural form. If the pH or the temperature of their environment gets too far out of whack, then they stop working. All right, so they are usually very specific to a particular temperature and pH. Enzymes act on a substrate, right? The substrate is the thing that they're doing chemistry on, and enzymes will usually end in A's. And often, not always, but often, What comes before the A's is whatever the substrate they act on is, right? So amylose is starch, all right? It's the technical term for that type of starch. Amylase is an enzyme found in saliva which will digest and break apart that starch. Literally, that means protein that acts on amylose. Enzymes are also, as catalysts, reusable. That means that they don't just catalyze one reaction, they can catalyze millions and millions and millions of reactions. Sort of like a factory worker could put together many parts. You take the factory worker away, the parts don't assemble themselves. But one factory worker could put together many different parts on a assembly line. And I know that we don't really do assembly line construction that way anymore, but it's a good metaphor. Enzymes are also very fast. One enzyme molecule can do millions of reactions per minute. So it's very efficient. Temperature, pH, anything that might cause a protein to denature can change the enzyme's shape. If you change the enzyme shape, you change its function. Salivary amylase, which we just talked about, works at pH 7. That's the pH in your mouth. Makes sense. Pepsin is an enzyme in your stomach that digests proteins. And what would you think a protein in your stomach would work at? Would it work best at neutral pH? No, it's going to work best at an acidic pH, around pH 2, because your stomach is acidic. Most human enzymes are going to work at the human optimal body temperature, which is 37 degrees Celsius. That's 98.6 degrees Fahrenheit, but you should get used to thinking in Celsius for this class, because we do a lot of stuff in Celsius. Make the body too hot or too cold, those enzymes don't work very well. This is, by the way, why you die if you get too hot. You start denaturing your proteins don't work. If your proteins don't work, you die. If you get too cold, your proteins don't work, you die. If you get too acidic, your proteins don't work, you die. If you get too basic, your proteins don't work, you die. If you move homeostasis too far outside of its range, your proteins don't work and you die. Usually when you die because something has caused your proteins to stop working. This is just to show you denaturation and renaturation. When you unfold a protein, you are denaturing it. Denatured proteins can sometimes be renatured by chaperones that will refold them back into their proper form, but that's actually pretty rare in humans. It's more common in bacteria and other things like that. But with humans, usually you just cut apart a denatured protein and then build another normal protein. But it is possible for renaturation to happen. The way enzymes work is enzymes have what's called an active site. That active site is the area that binds the substrate. So for instance, we're talking about sucrase, which is the enzyme that breaks sucrose into glucose and fructose, right? So it's going to cut apart sucrose, the disaccharide, into glucose and fructose. The active site of the enzyme is going to be where the substrate binds to. So it's going to have a very specific three-dimensional shape, and that specific three-dimensional shape is going to fit the substrate kind of like a glove fits a hand. And in the active site, you will usually have some special R groups somewhere in there that are going to perform the chemistry. So right here, at this juncture point, you're going to have an R group that allows the chemistry, the hydrolysis, remember hydrolysis is what separates polymers, that allows the hydrolysis to happen. It probably positions the water molecule in the right place. It will take the water molecule apart until the H and the OH, allowing that reaction to happen very quickly. And then the reaction happens, and the glucose and the fructose go free. All right? So here, in the initial state, we have the enzyme and the substrate. When the enzyme and substrate bind together, you have what is called the enzyme-substrate complex. And then the substrate is changed chemically, and you end up with the product or products. And then those products are released, and the enzyme can go grab another substrate. Some enzymes work perfectly fine on their own. They don't require any help at all. But some enzymes can't actually work. About 2/3 of human enzymes require, they can't do the chemistry on their own. They need some help. That help comes in the form of what's called a cofactor. This is a non-protein thing that the enzyme has to have in order to do the chemistry that it's doing. Cofactors can either be inorganic or organic. Inorganic cofactors are usually going to be ions, metal ions. They can be iron, copper, zinc, magnesium. Calcium is very common, sometimes sodium and potassium. Those are less common. But something that is required. For instance, iron is required for hemoglobin to bind oxygen. It can't bind oxygen on itself. It has to have that iron. Some cofactors are basically going to change an enzyme's shape, turning it on or allowing it to work. Some are going to be involved in the chemistry that it is performing. So organic enzymes, or organic cofactors, are called coenzymes. Right? So if what is required for the enzyme to do its job is an organic molecule that's not a protein, we call that a coenzyme. Coenzymes that you have to get as a part of your diet are what are known as vitamins. Right? So all those vitamins that you need to get, vitamin C, vitamin B1, B2, B12, all of those are all either things that are going to be coenzymes or things that you will make coenzymes out of. A big thing that coenzymes do is they help shuffle electrons around. If you're going to be making and breaking bonds, you have to be able to move electrons. And one of the things that proteins just don't do very well on their own is moving electrons. And so if they need to move electrons around, they often need a coenzyme that's going to allow them to do that. For example, NAD+ is a common coenzyme that is an electron acceptor. We'll talk about NAD+ a lot more when we talk about metabolism. And our last biological macromolecule, nucleic acids. Right? DNA and RNA. The monomer unit for nucleic acids is what is called a nucleotide or a nucleobase. The polymer, many nucleotides together, can be DNA or RNA. Those are the two forms that your body has used. Nucleotide structure, the structure of the monomer, is made of 3 parts. They all have these 3 parts. They all have a phosphate, which is a charged phosphorus and oxygen containing group. They all have a sugar, a 5-carbon sugar that is either ribose or deoxyribose. And they have one of 5 different what are called nitrogenous bases. DNA has 4 nitrogenous bases that it uses, and those are A, T, G, and C, which stand for adenosine, thymine, guanine, and cytosine. RNA also has 4 nitrogenous bases. 3 of them are the same. Adenosine, guanine, and cytosine are the same. But in RNA, the T, thymine, is replaced with U, uracil. The difference between DNA and RNA, or the differences between DNA and RNA, one of them is the substitution of uracil for thymine. The other is a change in the sugar group. DNA uses deoxyribose. That's what the D in DNA stands for. It stands for deoxyribose. RNA stands for ribose. All right? And what do you think the difference is? Well, the difference is 1 oxygen. So with DNA, you have your sugar, and every carbon has an OH coming off of it, except this one here, which has the base that I'm going to draw as a B coming off of it. And these carbons have numbers associated with them. So this is carbon 1, 2, 3, 4, and 5. And they are usually named with this little apostrophe coming off of them that we pronounce as prime. So we would call this the 1' carbon. This is the 2' carbon, 3' carbon, 4' carbon, 5' carbon. And the difference between DNA and RNA, this is RNA, by the way, or ribose. DNA is missing an oxygen, and it is specifically missing an oxygen coming off of the 2' carbon. So we still have the base coming off here. We still have a carbon here with an OH coming off of it, or a phosphate coming off of it in the case of a base. We still have an OH coming off here. But here, instead of an OH coming off, we just have an H. We're missing that oxygen. That's what makes it deoxy. This is D and A, or at least it is a deoxynucleotide, something that you will make DNA out of. As far as the chemical properties of nucleic acids, they're fairly hydrophilic. Some of them are very energy rich, particularly ATP, which we'll talk about more later on, and base pairing. And specifically, base pairing is what allows the DNA double helix to form. This is what allows your DNA to be used to store and replicate information, and it has to do with how the bases form hydrogen bonds. How are nucleic acids used in the body? The main way is information storage. All right? Your body stores all of the information about yourself in your DNA. All of the instructions for how to build a body, how to run a cell, when to do what, how to make what proteins, all of that is contained in your DNA. Your DNA is where it's stored, but that's not where it's used. It's used in the form of RNA. So DNA is like the storage form, and then when you want to do something, you go and make an RNA copy of the instructions. You bring the RNA outside of the nucleus into the cell, and then you use that RNA copy as instructions, right? And it makes sense, because if you've only got one copy of your really important information, you want to keep it someplace safe. And then you want to make copies of it for usage. but your master copy you keep safe. Second main function, energy. Now, you don't typically break down RNA and DNA for energy like storage or anything, but the energy currency that your cell uses is in the form of ATP and GTP. The A and G are the same A and G as are in DNA, right? Adenosine or guanine. The TP stands for triphosphate. So instead of having one phosphate, these have 3 phosphates in a row attached to them. And those phosphate-phosphate bonds are really energy rich. And this is really quick energy that your body can get at all the time. Right? You break down glucose to get energy. Well, how is that energy stored? Your body doesn't just like have energy sitting there. Your body's a chemical phenomenon. So all of that energy has to be stored in the form of some sort of chemical. When you break down glucose to get energy from it, what you're doing is you're making ATP. And then you can use the energy in that ATP to do stuff. And ATP is a nucleotide base, not a polymer, it's a monomer. But it is a nucleotide monomer. Control. There are hormones in the body that are made from modified nucleobases. Again, if you need to be able to make something really, really quickly, you want to be able to make it from something that you already have lying around, and you've got a bunch of nucleobases lying around already anyways. Last, chemistry. Now, we talked about how proteins can form enzymes, and those enzymes do chemistry in the body. And I said a few like kind of things like, most enzymes are made from proteins, but that implies that some aren't. And we're finding increasingly that there are some enzymes that do important things in the body that are actually made out of RNA. So RNA can also act as enzymes. RNA enzymes are called ribozymes. We don't know how important it is. We know that it's pretty important. There are some really important enzymes that are made out of RNA. But it's an area of active research, just exactly how many, what percentage of enzymes are RNA. We don't know yet. Many cofactors, coenzymes, are also made from nucleobases. They're not pure nucleobases, but you take the nucleobase and you modify it a bit, and that can be a cofactor or coenzyme that enzymes use to help them do chemistry. Now, their last function that I said that base pairing, right? This refers to the ability of DNA bases to specifically bond with a pair. Specifically, thymine, T, bonds with adenine, A, or thymosine bonds with adenosine. That just means in the nucleotide form. And it has to do with how hydrogen bond donors and acceptors line up. So for instance, in thymine, we have this right here. This is an acceptor. And this here is a donor, right? So this right here, this carbonyl group here, it can receive a hydrogen bond, but it can't donate one because it doesn't have a hydrogen. This NH can donate a hydrogen bond because it has a hydrogen. This NH2 group, this amino group right here, that is a donor. And this nitrogen in the middle here, that is an acceptor. So you can see the acceptors match up with the donors. Down here with guanine, guanine binds to cytosine, G binds to C, Here we have a acceptor, donor, donor. And on the cytosine, we have a donor, acceptor, acceptor. Note that thymine could not bind to very well to cytosine because they're both small, right? So this base here is a small base. This base here is a big base. You always have to have a small base bound to a big base. A small base is what is called a pyrimidine. A large base is what is called a purine. Large because it has 2 rings on it. And that has to do with the spacing. If you have 2 big bases, then they'll actually be forcing each other out. They'll be trying to occupy the same space, and that's not physically possible. If you have 2 small bases, they'll be too far away to form bonds. So you always have to have a small base and a big base bound together. And if you were to look at the thymine and try to match it up to the guanine, you see that, you'll be matching an acceptor to an acceptor and a donor to a donor, and you can't have 2 acceptors bonding. You can't have 2 donors bonding. If you were to try to bind this adenine and that guanine, you would have, a donor here, bonding up with this. Sorry, you couldn't have adenine with guanine. If you had tried to bind up adenine with cytosine, which don't base pair, you would have this donor matching that donor, this acceptor matching that acceptor, and they won't line up. So that's how the specificity works. DNA polymers are 2-stranded. So on one strand, one linear molecule, you will have one set of bases that have a specific connectivity. There's 4 bases, A, T, G, and C. So you might have it go one strand A, T, G, C. And strands, by the way, have a directionality to them, so we're going to read that way. The DNA molecule is anti-parallel, meaning that they run side by side but opposite directions, like a highway. And so the other strand will be what's called the complementary base, the base that one binds to. C binds to G, so it will go a G here. This G binds to a C, so we'll have a C. This T binds to an A, so we'll have an A. These 2 A's bind to T's. So the complementary strand, what makes up the other side of that double helix, runs the opposite direction because they're anti-parallel like highways, and it will read G, C, A, T, T. You'll probably get more into the structure of DNA in your intro biology course if you haven't already. We'll probably cover it a little bit more in this class, but that's about the amount of detail that I have to go into the structure of DNA. I do want to touch a little bit on ATP because it is something we're going to be talking about a lot. And ATP is the same A, so this is that large base, that adenosine, right, bound to a ribose. It's a riboRNA base. And then instead of having 1 phosphate, we have 3 phosphates. 1, 2, 3. The one that's closest to the molecule is called the alpha phosphate. The one that's in the middle is the beta. The one at the end, the third one, is the gamma phosphate. This bond here, this beta-gamma phosphate bond, is very high energy. That means that you can store energy by putting the gamma phosphate on. And then that energy is locked up in that bond. And then if you break this bond, you release that energy. We'll get in a little bit into how that works when we talk about metabolism, but this is how your body reversibly stores energy in chemical form. It's very, very fast. fast. All right, those are the 4 biological macromolecules. You should know what we went over for each of them. So for each of them, you should know what the monomer is, what the polymer is, the importance of the structure, and the functions in the body. And for proteins, you need to know the 4 levels of structure, at least basically what they are, as well as what the main force holding them together is: covalent, backbone, hydrogen bonds, hydrophobic interactions for the first 3. You don't need to know the main force in quaternary because it's the same as tertiary. For DNA, you need to know what base pairing is. You don't have to memorize all that stuff about donors and acceptors, but you need to know that A bonds to T, G bonds to C. You need to know alpha, beta, gamma phosphate groups and which bond is broken. And in ATP, when you're using energy in ATP, which one is made or broken. And you need to know you need to know the differences, the chemical properties of each one. So you need to know chemical properties of sugar, particularly with lipids. I want you to know that they are hydrophobic and that there are 2 forms, fatty acids and cholesterols or steroids. All right, so that is the ending of, not exactly the ending of this chapter's materials, but that's the ending of this section on carbon chemistry.